Hi everyone! Today we're going to learn about electrophysiology. So, what is electrophysiology? In a nutshell, neuronal electrophysiology is a study of the electrical properties of cells and tissues in the nervous system. This is a great way to study neuronal activity, as most neurons in your body communicate with electrical signals. We're going to talk about how neurons communicate with electrical signals, the techniques that electrophysiologists use to study these signals, and why it is important to understand the electrical activity of the nervous system. Let's start with how neurons communicate with each other. All neurons communicate through synapses, which are small junctions between nerve cells. Synapses can be chemical or electrical. In chemical synapses, small chemical signals called neurotransmitters pass from one neuron to another and transmit information. In electrical synapses, ions pass from neuron to carry information and commands. These ions, usually potassium and sodium, create something called an electrical potential. One type of electrical potential is called the resting potential, which occurs when there aren't any ions passing across the synapse and the neuron is at rest. Another type is called an action potential, which occurs when the neuron is stimulated and ions flow through the synapse. Think of action potentials like an email. When you receive information from a source that you want to share with someone else, you could send an electronic communication in the form of an email. The email carries the information from the sender to the receiver. Ions work the same way. In a synapse, ions go from one neuron to another, carrying electrical signals that take the form of commands. Electrophysiology techniques study and analyze these electronic signals that neurons send to one another in order to decode the messages sent throughout the nervous system. So, how does electrophysiology work? Let's look at the most common techniques. Firstly, we're going to look at intracellular electrophysiology. This means we're taking recordings from inside individual cells. The first technique is called patch clamping. In patch clamping, a micropipette is brought into contact with the cell membrane of a single cell. The micropipette is filled with electrically conductive liquid, and the micropipette tip encloses a small area or patch of the cell membrane. The membrane potential of this area can be recorded. Patch clamping isolates this membrane patch and allows for analysis of cell membranes and how the membrane responds to changes and how it differs between cell types. The second technique is called the sharp microelectrode technique. This technique involves piercing the cell membrane with a sharp and small micropipette so that the electrode can come directly into contact with the interior of the cell without affecting the ions in the cytosol. The micropipette opening is far smaller, so there is no mixing with the cytosol as seen in patch clamping. The tip can be filled with different dyes to mark the cells that recordings are being taken from. However, as the cell is being physically speared with a tiny glass spike, permanent damage is done and recordings cannot be taken long term. Our third technique is called voltage clamping. Voltage clamping involves using one intracellular recording electrode and another electrode that delivers voltage. While one electrode records the membrane potential, the other electrode can add voltage to the cell, which stimulates the cell membrane and changes the membrane potential. This clamps the membrane potential at the desired voltage level, where it is maintained while recordings are taken. Voltage clamping allows researchers to observe cell behavior at different voltages, also using visual recordings to see if the structures inside the cell are still functional. Next, let's look at extracellular electrophysiology. This means we're taking recordings from the exterior of a cell. While intracellular electrophysiology is more accurate, it can only examine one cell at a time. Extracellular recordings can look at multiple cells at once. The multi-electrode array, or MEA, is a device that contains anywhere from tens of thousands of microelectrodes, which act as interfaces between cells and circuitries. Typically, cells are grown on the MEA, and their physical contact with the electrodes means that extracellular recordings can be taken from anywhere in the cell population. Cells that are spontaneously active, such as neurons, can be recorded non-invasively over the long term across a whole population. Why is electrophysiology important? Well, studying how the electrical environment of the cell can affect the flow of ions, and thus the functionality of the cell, is studied widely for its applications to disease prevention and remediation. Additionally, knowledge of electrophysiology techniques, as well as the common complications, can lead to higher quality data generation. Finally, combining electrophysiology with certain visual techniques has contributed even further to the field of neuroscience and maintains the status of electrophysiology as a vital technique to understanding our human biology.